We can get started. Good afternoon, everyone. Thanks for making the trek over in this amazing weather. But who are we kidding? We live in South Bend. It was a choice we all made. But Amarda daren't. So I'm sorry that we're greeting you with this weather, Amarda. But it's a pleasure to welcome Amarda Shehu from George Mason University, who has the most titles that I can have ever come across. Professor of Computer Science, Associate Dean for AI Innovation, Associate VP for Research, Co-Director for Center for Government Cybersecurity. I have to read it. Co-Director, Center for Commonwealth AI, and Founding Director. But more importantly than all these titles, he's an exceptional scholar and scientist, having published more than 180 papers, several awards, NSF Career Award, Beck Family Presidential Family Award for scholarship, and just and significant impact coming from a research. And more importantly, I feel, I have known Amarda's work for quite a while. As you know, we are now talking about AI for science, AI for scientific discovery. She's been doing it, talking about it for way longer before it became a thing. She also served as an NSF program manager and was instrumental in directing several programs, including the Harnessing the Data Revolution, HDR Data Science, and a couple of us are funding from that program as well, Amarda. So thank you for doing all that for us. And she was also did the COVID-19 response when the NSF was running that as well. And one thing I've gotten to appreciate and respect about Amarda is we were recently on his writing a report for NSF on AI for science. And when someone said, okay, we need to write this, there's about a dozen of us in the room. The only one to raise her hand was, I got it. I can do this in 30 minutes. I'll write a call for proposal in 45 minutes, then move on. So this is who Amarda is, an amazing scientist, scholar, and an amazingly kind and collaborative person. That's what leads to this. So Amarda, thank you. We look forward to your talk.
Thank you. Thank you. It's an art to know when to say no. And I haven't mastered that yet. So I'm looking to streamline some of these one day into one position, only one. But this is going to be a talk about research. And since this is my first time in Indiana, not just at Notre Dame, but in Indiana, I thought maybe this is a good opportunity. And since there are going to be students here, to give somewhat of a broad overview. And whenever you try to do that, it's very hard to pack long time research into something digestible where you can see a logic model, right? I'm all about why are you doing it. But if I had to summarize sort of the research in my lab now and then, it would be somehow we maintain two thrusts or two legs. We've always had one leg in foundational research. So caring about algorithms, I consider myself an algorithmic person. I started in theory, I started in logic, and then I left that and I said, I'm gonna go and do my PhD in AI. But we still do a lot of foundational research in AI machine learning, and you will see kind of glimmers of that during the talk. I'm not gonna bore you. And then we keep another leg in applications. We very much want to do use-inspired research. That is mainly because I've always, since I was, I think, in high school, wanted to be a scientist and not a computer scientist. I accidentally ended up in computer science. Mainly it was because that was the only place where George Soros gave me a scholarship. Otherwise, I wouldn't be able to afford college. But the beauty of these sort of two loves, two passions, is that they feed into each other. Very often the problems that we go after in the application side, they're the ones that challenge us enough and tell us we don't know the heck what we're doing. So they really raise very interesting questions that then allow us to make foundational contributions in advanced computer science research. So I'm very much about the why. It needs to be a reason why you're doing this. There is a purpose. So computing for a purpose, that's me. It's a very biased view, I do admit that. And we've been fortunate enough to be funded by different funding agencies over the years. And I do bear the scars of those on my back. It's too many rejections, and these are only the successful, the wins that you see. Okay, so going back to that scientist that I've always seen myself as a scientist. So to all the students here, did not enjoy my PhD in my first few years. I really didn't see the point of it. I was like, I don't know. I see these guys around me cracking jokes, obscure jokes that I don't understand, not a part of these. Why am I doing this? Why was I asked to do this? But I had a liberating moment, believe it or not, when I stumbled across a paper given to me by a professor in chemistry, written by Sir Alan Turing, who in computer science, we call him the god of computer science, but apparently he was a polymath. So he wrote papers for other domains. So he started to venture into chemistry and biology, and he started to think about biological systems and how they evolved. How does tissue become an organ? What are the states through which matter goes through? And in reading that paper, he basically raised the question that has been raised for millennia by curious humans, always evolving. How does the form of matter determine the function of matter? It's a very mechanistic question, right? It's a question that says, if I understand what you look like, then I can try to understand what you could be doing. Why does a hammer look like a hammer, right? What does a hammer do? What does it being looking like a hammer allow it to do? So this is a question that you find instantiated in sciences. So I kept finding it more and more and more as I started reading papers, and it became the calling of my heart in my scientific career. It was the guiding principle, and I found a very nice medium for it because the problems that I was drawn after, I didn't want to write sorting algorithms. To be very honest with you, I didn't want to make them faster. I just did not see the point for it. But the point I saw was that if I could address even a tiny, teensy part of societal and health problems by making fundamental scientific advances in computer science, then that was good for me. That was good enough for me. So I latched on to this question, how does form determine or govern function? And the very first moments when I started seeing instantiations of it were on physical systems, robots, and biological systems, molecules. And so that question became a beacon towards something that puts together all my research, gives it a focus, and it is building or learning functioning coding representations of form. So whenever you want to understand something about form, the shape, the structure, we call it by different names, configuration, conformation, state. The moment that you want to write up some code about it, you make a choice of representation. What detail you leave out and what detail you put in. You want to leave enough detail that allows you to capture what you're looking for, the function. But because it's an approximation, the moment you start putting it in words, you're making an approximation. If you understand anything about how we think and how we talk, you're only building, I call approximative representations. Again, I found solace in none other than Aristotle, who said it is okay to be content with approximation. Chemistry students, physics students, they understand it very well. They talk about first order approximation. Let's think of it linear for computer scientists. Second order approximation, let's think non-linear for computer science, right? So you get my point. So that was my beacon and the computational focus of my research. So representation learning in my lab over the years, it's not possible to really scrunch up everything together, but it has undergone an evolution from no data. We don't have any data, which is hard to believe these days. Now we have some data, and now we have a lot of data. All right, so if I had to move it into the pre-data era, into the little data era, and then in the lots of data era, these would kind of be the three epochs of research in my lab, again, guided by representation learning. All right, so here is my logic model. I tried very hard to kind of make sense of everything that we're doing. So you will hear representation, 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 the same way that the real estate agent tells you location, location, location. Now, there are two arms that we keep. One is discriminative, one is generative, all right? And most of this talk is about generative, but I will hit on discriminative too. In discriminative, I'm talking about prediction tasks, so form to function. If I tell you what it looks like, can you tell me what it does? All right, prediction of something measurable about function. In the little data stage, into the lots of data stage. Generative, I care about capturing many forms of this system. I tend to move around a lot. So if you want to capture how do humans move, Can you do that from one form? Can you tell me all the different forms of humans, right? Can you give me the ensemble of forms? Can you sample the state space of humans, right? We talk about generating text and generating video. I'm not going to tell you about that, though one of my students is in NLP. I'm going to tell you about generating shapes of things, structures of things, of robots, and of molecules. The focus very early on in my career was on complex objects or articulate objects, objects that basically have many joints. These are your joints, right? They don't move so much after 40 years old. And the links that connect the joints, right? So everything that I was looking at, that's that mechanistic curves. Everything you look at, you're tracing where the joints, where the links, right? Every object that I focused on had to be a modular object that operated in the presence of constraints. What that means is that the shape of potential configurations of that system or forms of that system is highly constrained, which is great for computer scientists, because then you can talk about constraint optimization, right? You're not like, I don't know, this space is so dimensional and nothing really makes sense. You're going after very specific things. And if you can capture those constraints as objectives, then you can talk about loss functions. Now they go by the word loss functions. We used to call them just objective functions back then. All right. So the kinds of systems that I started looking at were these. These are known as protein forms or protein structures. And I was just focusing in the beginning in bringing them alive, giving them some diversity. We knew, for instance, that very specific portions of these, I don't think this works, but very specific portions of these are movable. If you go and you try to sort of take snapshots of a molecule, and pardon to all my chemistry faculty here, you kind of capture them in a snapshot, but next time you maybe take a picture, they look a little bit different because they're always in motion, okay? Life is in motion. They undergo thermal vibrations. So what I really wanted to capture was this ensemble of equilibrium forms. So ensemble of shapes of things that operated at equilibrium, which entailed certain energetic constraints. And first I started with small portions, loops that you see here. From one loop, so one data, think of it, can you generate an ensemble that is energetically feasible? And then from an entire chain of a protein to replacing that one structure with an ensemble of equilibrium conformations. Now, there was very little data. I couldn't throw this up to some generative deep learning. So what I had to do is I had to understand deeply about force fields. Computational chemistry folks know about this. So the objective function, what is it? What it is you're trying to optimize so that these forms are physically realistic. And I had to make analogies with robotics where we understood how to do kinematics, how to compute forms fast, and how to compute changes to forms fast, and how to do inverse kinematics, which was important for things like these loops, because you wanted all those shapes to be hooked at the beginning and the end to the rest of the protein, and that's an inverse kinematics problem. So there was a lot of thinking about what are the parameters, variables, the degrees of freedom. These are interchangeable depending on the community where you are. You may ascribe to one or the other. The idea was, how do you control the dimensionality of the search space, the space of all potential forms? 99% is junk or not relevant to you. How do you find that 1% that you care about, that satisfies all the constraints in which these systems, these biological systems in this case, have to operate? Okay, if I show this picture, how many people explicitly or implicitly feel that they know what this picture shows? Okay, thank you. Some of you do. All right, so
That's impressive.
This is a beautiful picture that I used more often, now not so much, but it basically shows, and I'm going to go very quickly, it basically shows how proteins are formed. Here is the Mickey Mouse thing. Here, this organ here is the ribosome. And so messenger RNA goes inside there, and transfer RNA. These little things here that have these pink or purplish spheres that are supposed to be amino acids. They bring them in a certain order per the code of messenger RNA. And so this chain gets formed, gets linked, but it doesn't stay loose. It actually assumes a specific shape, a specific form that we call the native form. Okay. When you look at this picture, a computer scientist says, and many people at DeepMind now say protein structure prediction, done that. Okay, so this picture is supposed to illustrate a very important problem for molecular biology, protein structure prediction, which basically says, if I have a slide, I have it later. I have this one, okay, which basically says, if I give you the sequence of amino acids, those building units that come together and hold hands for the chemistry, can you tell me what is the shape that they take in 3 dimensions? Because many proteins, when we find them, when we experimentally resolve them, we find them in some particular 3-dimensional shape that we call the native shape. And this seems like a well-defined problem, but there's a reason why it's a well-defined problem. And the reason is because of multiple what are called disruptions in biology. This is a highly biased projection or view of disruptions from Charles Darwin that says biology is quantitative. Yay, great, can do something there. To Watson and Crick, who cheated off Rosalind, but they got the Nobel laureate and nobody talks about her anymore. But I do when I get a chance. They gave us the double helical structure of DNA off of her X-rays, by the way. And then we have Anfinsen in the 50s and 60s who said that, you know, those amino acids, when they get linked, they have a memory. There's something encoded in them that drives them to the 3-dimensional shape. So he said that, I mean, he did many experiments to tell us that, but basically that was a charge for computer scientists, mostly physicists at the time, that there's logic, there is method to the madness. And if we can figure out where that memory resides, we can reproduce computationally that 3-dimensional structure. Perutz and Kendrew, they gave us, through experiments, the crystal structure of hemoglobin and then of many other proteins. In Scientific American, they write a story we report on. It's like goosebumps when you read that story. It was like, you know, like landing on the moon kind of thing. The first time we saw 3-dimensional structure, it was a little bit thick, resolution wasn't so good, but we saw a 3-dimensional structure of hemoglobin. And then we have these guys. These were primarily physicists, I would say. And here I'm only showing Karplus, Michael Levitt, and Ariel Varshel. And they got the Nobel laureate for their work in computational chemistry, these 2 on force fields, and this one on molecular dynamic simulation that would kind of simulate the wiggling and the jiggling of atoms that Feynman talked about, and told us that there are computational ways to simulate how molecules behave and to see how they transition between different forms, different shapes. The Nobel laureates left out McKeven and my favorite Sharaga, because Sharaga was the one that said optimization. These other guys didn't really talk about optimization. They were physicists, they talked about simulation. Sharaga said, this is an optimization problem. If we have a function that captures the interaction, we can go after the minimum of this function. We just have to be smart about the algorithm. He was a little bit wrong, okay? But he motivated scores of scientists, including me. When I read this paper, I said, that's why I'm doing this thing that I'm doing. And then a lot of us did a lot of work in this space until about December 2020, when I was at CAS, and I saw the presentation by the DeepMind team, and I said a word that I cannot repeat in public. Okay, but basically now that 50-year-old challenge is considered solved. Okay, why is it considered solved? Because a lot of us did a lot of work to set the stage for it. So I'm not putting my work, I'm putting the work of my dear colleague, Jin-Bok Su, and collaborator, who gave us a new approach to thinking about how to put together that 3-dimensional structure. Jin-Bok Su had the idea, residents were starting back then, this was like 2018, 2019, but he had this idea since 2017. And he put together a resident that could predict from sequential features. So basically over the amino acids, pairs, which pairs of amino acids were in contact, were close to one another. Didn't give us the 3-dimensional structure with the Cartesian coordinates of every atom, but give us a good idea. Think of it as pairwise distance geometry, right? And it would be a very easy problem, no difficult problem, to go from which pair is close to, which amino acid is close to which, to get to actual structure, but he did not do that. You know who did that? The DeepMind folks. They went to pairwise distances early in their AlphaFold version, and then in their AlphaFold 2 version, they completed the story and gave us 3-dimensional structures. I'm not going to go there. A lot of bells and whistles here, but I do want to say that a lot of the people that work here were postdocs of faculty that developed these ideas over 20, 30 years. So it's not that they came up out of thin air. They stood on the shoulders of giants. So I want to tell the students that the research that you do here at the university is what is actually powering a lot of those advances in industry. They just have way more money to prototype the heck out of their systems. Okay? You have to be smarter. Okay, so AlphaFold 2 is really good, but I want to show you what it cannot do. It cannot do this. It cannot build dynamic pictures of proteins. It can give you one structure, it could give you 5. You can ask for 5 structures, you can tweak with it, but they're mostly similar. Because to be able to get that one structure, there has to be a lot of bias in that neural network to get to that one structure, right? Neural networks are very good at that. You have to do inference. So it doesn't tell you how the chain folds. That physical process is very important because some proteins, some faulty proteins don't get to that structure, they get to something else, right? It's not sensitive to mutations. If you tweak one amino acid, which is what you find in disease, it's still gonna give you the same structure. It's gonna give you the wrong picture. But don't take my word for me. I'll give you an example that really showed structural plasticity in action. So I know we don't like to get reminded of COVID. Last time I missed this talk is because I got COVID. So I had to cancel my September trip. But what COVID showed us is that structural plasticity, or the ability of molecules to be dynamic, is inherent to its function. So the spike protein that gets so much talked about, and I'm showing here, has a domain, a portion of it, let me not define. Most of the time, 90% of the time, it stays in a particular form or it evades our immune system. 10% of the time, it opens up. It's not working, I'm sorry. Closed, open. When it opens up, when it changes form, then it's able to bind. That form allows it to bind AC2 receptors in the blood and in the lung, and then bad stuff starts happening. So if you don't understand dynamics, you don't understand mechanism of action. If you feed this thing to AlphaFold 2, you're gonna get the closed form. And you just say, we're safe. Okay, whatever, this thing is there, it's not interactive. You miss the fact that it can populate with low probability, but it can populate other different forms. So the picture of molecules is not a static picture. If you have, again, pardon to my chemistry and maybe statistical mechanics faculty in this room, but it's a probabilistic question. At any point in time, if I ask what is the form of this protein, you can say, well, there are many replicas of this protein in this cell. And maybe 90% of them will be in the closed structure, 10% will be in the open structure, or maybe the probability distribution looks different. So the question of dynamics really comes to what's that structure space and what's the probability with which these things assume particular form. Okay, but most computer scientists don't want to talk about dynamics. You guess why? It's a very difficult problem. It's a very upsetting problem. You make very little progress and you can get very little money from funding agencies to fund a lot of work. Okay, it's really difficult. The upside is, though, very little competition. Okay, so that is one of the reasons that I focused a lot on generative. I didn't call it generative back in the day, like 10 years ago. I told, you know what I called it sampling. I was sampling the conformational ensemble because we didn't say generative back in the day, right? And I didn't work in structure prediction because even before AlphaFold came out, there were huge labs with 30 PhD students, 10 postdocs that I could not compete with as a meager assistant professor with 3 PhD students. So I said, we're going to do the more difficult thing that nobody wants to do. Okay, so sometimes it's useful to do the more difficult. Okay, so the more difficult thing, the inspiration that we got, we were not doing molecular dynamics, we were not following Newton's laws of motion because they take forever, right? And a lot of computational chemists will do that, so we're not interested. We were taking idea from robotics. We were looking at robots operating, this is real schematic, operating in a configuration space, trying to go from one configuration to another configuration while avoiding collisions with obstacles. For proteins, the picture was much more different because the protein has hundreds, thousands degrees of freedom you have to contend with, and there are no solid obstacles. There are energetic obstacles, right? There are peaks and then there are valleys. And so I wanted to see, for instance, even if you gave me 2 structures, let's say, of that spike protein. You said here is open, here is closed. How does it navigate? How does it transition between these two? You have to carve a path in this, what I call the energy landscape, right? It's a structure space that has an additional dimension that is the energy, right? For folks in deep learning, your loss function, okay? Your loss landscape that you call it. Okay. So we developed a lot of search algorithms. My heart is still there. Even though we're doing a lot of deep learning, my heart is still search and optimization, my first love. So we developed a lot of tree-based algorithms where given two initial conditions, call them A and B, two different structures, we would try to find an optimal, but we couldn't make guarantees. So let's say near optimal path. The path would have intermediate structures, so we would have to sample structures and connect them. And there are different ways in which we can do this. We would take ideas from robot motion planning and adapt them to very high dimensional spaces. Or in order to improve our guarantees and improve on the optimality, we will generate roadmaps or graphs. So we will try to capture the connectivity of that energy landscape by growing a roadmap or a graph. Again, when all we had were two structures, and at times it seemed like really difficult. But we made a lot of interesting contributions, even foundational contributions, because we realized that when the space is so big, you don't know where to go. You can get lost. You need information about where you've been. You need memory. But how can you have this memory? So we introduced discretization layers. So we introduced an energy discretization layer, and we would bin it because we wanted to remember how many structures have we sampled there are in a particular energy bin. And we introduced a discretization layer over the configurations themselves. We would summarize, encapsulate these structures with a few coordinates, things such as the distance to the centroid, distance to the point that is closest to the centroid. So some features, we were trying to design features for molecules so we could embed a grid. And this way we had memory. Anytime I wanted to say now from where should I grow this tree, in iterations, the tree or the roadmap, I would say, well, when I figure out which conformation or which node from which node in the tree to grow, I really want to do it by the statistics, because now I can tell which energy level and I can tell which other structures are in this discretization or the projection of the conformation space. I would have a probability distribution function, a penalty that would tell me you have too many things here. You've been too many times in this region. You are wasting your computational time. Or look, this is a good energy layer, or you really don't want to waste your time going up in energy, but you do want a non-zero probability not to get stuck all the time at a low energy level because we don't want premature convergence. So there are a lot of games that we were playing. It was a lot of fun. We called it adaptive search, not adaptive learning, adaptive search guided by these projection-guided discretization layers. We applied these ideas to roadmaps too. When you bring it to roadmaps, then we had another idea. When you only have a limited computational time, which now we call compute. I would have trouble with compute because it's a verb. Name is not a noun, but anyway, limited computational time. Nowadays, a lot of the students will think, throw it to my 80 GPUs, yeah? Limited computational time, where do you want to spend it next? So we would build what we call fuzzy PRMs, meaning I really don't have the structures here, but I think they're close enough. They should connect without going through a barrier, but I'm going to give it a low probability. So next time I have freed, I'm like, okay, I have time in my budget. Which one wants to? Which one wants to be realized? We would pick one of these fuzzy edges based on their probability. And so edges, for instance, where we had spent a lot of time before, not made much progress, they would go down in the queue. They will not be picked up. So we had to do a lot of tricks because the space was really difficult, very high dimensional. We connected with Markov state models because we realized that these roadmaps are stochastic roadmaps. We could associate probabilities. Whenever you have energies, you can associate probabilities in physics. And so these probabilities are basically transition probabilities between states. And so we could obtain from the roadmaps, we could obtain summary statistics, so kinetic measurements that then we could connect with experimental measurements to basically tell us how badly we're doing, or yay, we have a paper. Okay, but let me show you the fun part. What my students really loved was producing these pictures that basically show transitions mediating two structures of different proteins that are very far apart. You may not appreciate how far apart 13 angstroms is, but ask any, I don't know, chemist, computational chemist student here, and they're gonna tell you, it's really far away, man. It's really far away, or 16 angstroms apart. But notice there's something missing in these pictures. Do the chemistry students notice something missing? There are no wiggling and jiggling of the atoms. They are moving like robots, right? All the detail has been lost. Because if I also had to put those individual bonds of those hydrogen atoms, the dimensionality is practically intractable. So we were reducing, remember I said approximative representations. So we were throwing away the atomistic detail and only focusing in this case on backbone with some approximations of side chain atoms for folks that know what I'm talking about with side chain atoms. The point I want to make is they look beautiful. We got a lot of high publicity for this, but very cognizant that these are approximative. We don't have detail on the atoms, the jiggling and the wiggling of the atoms, okay? All right, so discriminative, I just want to give a couple of examples without going too much. We did some work on trying to figure out, okay, what does this thing do? What does that thing do? So we did some work on DNA. One of the cool things that we did is we didn't go the route of, let's try anything we can think about, features, compositional features, and then throw them up to some, I don't know, SVM, they seem to be pretty hot back in the day. We didn't do that. That was very boring for us. So we wanted to understand the problem. So what I'm showing to you here is a piece of DNA and a promoter protein. It comes and it latches onto, it binds, but it has shape. It's a three-dimensional thing. So obviously it has to recognize regions that are far apart, right? This is a big table. But if I come with my hands, I'm recognizing two specific portions. So we realized that if you want to capture features, you want to capture maybe correlational features. So we came up with the idea of motifs. We want to be able to construct motifs. Feature engineering, feature construction was pretty hot before deep learning. It just remembers that. So we're like, okay, we understand the problem. We understand, remember, function encoding representation. The function puts the constraints. I just have to be smart to capture this in my representation. So we wanted to capture distal features, like a certain motif here and a certain motif here, which we represented as parse trees. And we realized we could use that beautiful Boolean algebra here. Because I can say, look, I can really represent a feature as a motif at this position and a motif at this other position. This may be a signal of, or what it means to be a promoter, right? But then we realize, but we may be biased, right? We understand something. So we could put many more because that and can become an or a not, right? That I can go away. The length of the submotifs can change. So if you are changing this, this is your search space. This is your feature space. So then we came up with, how do you explore this? And we used genetic programming. That everybody told me it's dead back in the day, but I have a habit of ignoring people. So we use genetic programming for feature space exploration. Genetic programming was being used to design programs, computer programs. These look like computer programs. So once we made that analogy, we used genetic programming. What was our objective function? A proxy machine learning model. Even naive was good enough on a small training data set. And it's actually small for like thousands was like, woo, so much data. Okay, other things very similar, protein structure, what does it do? We figured out we can cut it into fragments, so we can figure out these are fragments that are used. We find them in many different proteins, so we can count up. And there are about 200 different fragments we see across protein structures. So in a particular protein structure, we can say, yeah, we see this fragment, but we don't see this fragment. So this looks very much like term frequency, DF-IDF, for people that have done text mining. So again, we're building analogies. We realized, oh, we can look at proteins as bags of fragments / bags of words. Again, approximation. This is the first order approximation. We're not capturing correlations between fragments, right? But it's an approximation. We used LDA. Again, it was very hot in the day, still remains, I think, in the social sciences, to bring it up from 400 dimensions to only 10 dimensions, so 10 topics representing proteins. And then this really helps with the curse of dimensionality. SVMs don't do well when you have a lot of features. For those of you that may be taking introductory machine learning before deep learning. So reducing the dimensionality was important, and it gave us some of the best performance in the day. Okay, and several other problems. But the point I want to make across is when you don't have data, you have to understand the problem. So that then can guide you with what it is you want to capture, right? Or maybe how you can capture that. So you really have to love what you're doing. At least another way that I'm saying. The beautiful thing is we were working in an application domain that seems very far removed, but that gave us a very interesting foundational advances because we realized we could evolve features for promoter recognition, or we could evolve kernels to improve the performance of SVMs without any interest in any particular application on it, right? And then we did joint feature and kernel evolution to improve performance of SVM. So it gave us some neat machine learning papers as well. And this was a side effect. If I had not been involved in this, maybe I wouldn't have appreciated, how to construct more complex features and how to sort of navigate a complex feature space. All right, back to generative. Life is in motion, everything's in motion. So surely even when you ask the question, what does this thing do? It is not quite correct because often you cannot tell it from one shape. Remember the spike protein that I used? You have to have a picture of the dynamics. But before you do that, you have to be able to see those alternative shapes. If you don't see them, you can't even answer the question of what the mechanism of action is. Now, the beauty is that this is not just in biology. The examples I'm picking, there's work that we have done in structural engineering for bridges. You're like, what does a bridge have to do with molecules? A lot, man, a lot. You want to design better bridges? That's an optimization problem, right? How do you design better bridges? It's an articulated object. Right, and my civil engineer colleague would probably tell it much better than I do, but bridges are not static things. They are dynamic objects. They have to be dynamic objects. So there are a lot of very interesting ideas that kind of bridge across scientific disciplines, from robotics to structural engineering to material science that I've not talked about, how to design new materials. We've done some work in that too. The point that I'm trying to make is that in all of these different domains, it's very important to capture, some people call it emergent behavior. I'm not sure about that. It is like alien stuff to me. I want to be very humble and say to capture the dynamics, okay? And then maybe from the dynamics, I can tell you what it does, but just one step at a time, okay? So how do you capture the dynamics when you have little data? You don't have a lot of data. You have little data, okay? What's little data? Let's say 87 structures. Like I have specific numbers from projects, 87 forms. And I want you with these 87 forms to reconstruct for me the energy landscape of a particular protein that my colleague who studies cancer really cares about. And I do too, because I'm aging. So I call it knowledge and data-driven AI, okay? Because before I was just doing what I call classic AI, right? One form, lots of understanding, search and optimization. So, okay, what can we do here? I just want to kind of hit the home a little bit, bring home the point. On the left is an important protein for a bunch of human cancers, HRAS. It switches between two states. Okay, in one state, it tells the cell don't grow anymore, and in the other state, it says grow. So a lot of tumor, it's implicated in a lot of tumor growth, but suddenly this transition is disruptive in different ways. And I say in different ways because cancer is a very complex disease, and even what I'm saying makes me uncomfortable. That's how complex it is. But what that motion corresponds to is a particular path in the energy landscape of HRAS. It's a particular path drawn in yellow. And you might ask, what are those other deep wells that I see? Very good question. That's what we want to know. What else hides in those? And while all we can do maybe is capture it in these two states, what else is there that we don't yet see? What states hiding there? And do these other things also transition between those? And how does that impact healthy Or how does it impact disease? Does it become a factor in other mutations? I wouldn't know if I don't have a sense of the energy landscape of the map, basically, in which HRAS operates. And this is true for many proteins. So how do you expand it to, building the full state space so that then I can ask the question, I want to go from here to here, you tell me what is the most optimal path, the most realistic path, right? I don't want to have to limit it to only two particular structures. Build me the city, right? Where the structures are important landmarks, right? And tell me, how do I navigate between these landmarks? That is the connected energy landscape. Once I have this, then I can support a lot of queries, what and how and when as well. And then answer the questions, when there are perturbations to the systems, how does it affect these transitions? So I can try to get at the molecular level understanding about the disease. Now, disclaimer here. Yes, Diana, go ahead.
Sorry for interrupting. I had the same question on one of your earlier slides, and I'm dying to know the answer. For these yellow spots that you're interested in, do you actually obtain 3D coordinates of
Yes, we
evidence?
do. I will show you.
Where's that data?
I will show
I
you.
can't find it.
I will show you.
She said she would show you, she wouldn't give you.
Oh, I'll give you.
I'm teasing
I've
you.
got a lot of things on you. The data is a lot. Like, I'm hating it. At my university, they're like, you still need this? I'm like, yes, I still need this.
She's the last university we're getting rid of data in 17 days.
That kind of data is really hard.
I just
impossible
wanted
find.
to make a disclaimer here. What we can do in computation is a very small picture of the actual answer. Any disease, whether it's cancer or other disorders, is a very complex thing. These molecules, these proteins, don't exist in isolation. Okay, they interact with other proteins. We can't model all of those. We're making an approximation simply by saying, I'm just going to focus on this protein. The moment I say that, a lot of oncologists that I work with, that I know of, say, wait, what? That makes no sense. We know the environment is rich, and the ecosystem for this type of lung cancer is different from that other type of lung cancer. So they bring a lot of complexity in, but complexity, we don't like complexity in computer science. So hold it out, I can't even deal with this. I just want to focus on one thing. So I just want to tell the students, don't think like she solved the world. Far from it. I'm focusing on one molecule, and every disease is so complex, and we're not there yet where we can model the full cell, the full tissue. The moment that we can do that, then we may really have a picture about disease, okay? Just to basically tell you in the words of one of my students, biology is messy. That's what Brian used to say. Okay, so Tiana said, where do you find these other structures? Okay, so we had a lot of questions. Like, these energy landscapes, these proteins are big, thousands of dimensional space. Can we leverage some data, little data again? So we started asking the question, could we leverage deposited structures? So for HRAS, we went to the protein data bank, and not just for HRAS, for other proteins, but I'm only choosing HRAS here. We went to the protein data bank and we said, how many labs have deposited structures of HRAS? And they have deposited, but they're not the same protein molecule. They differ, they have mutations, or they're engineered in order to be more stabilized. Some have oncogenic that are known are oncogenic mutations. So we collected about 87 different X-ray structures, okay, of good to okay-ish quality. We had to sometimes really compromise on the quality in order to increase the amount of data to 87 structures. But these structures were not of the same sequence of amino acids. They were not of the same protein. So the question I had was, but can I use them still? I know that they look slightly different. They look like slightly decrepit versions of me. One is missing a limb, one looks a little bit crooked, but still they're variants of me. Can I use these if I want to build the energy landscape of me or of any of these particular variants? And my inclination was right. I found the theoretical basis because I was collaborating with a molecular biologist, Ruth Lucinol, that was really trying to bring more forward in molecular biology the principle of conformational selection or population shift. So in terms, Ruth's, I call it Ruth's theory, but she hates that. Conformational selection says structures don't just come out of the vacuum. If you tell me, if you show me the structure space of HRAS in the healthy form versus HRAS in some oncogenic form versus HRAS in some other disorder that's not oncogenic, each of the structures for each of these exist in each of the structure spaces. The only thing that is different is the probability with which any of these particular variants of HRAS populate those structures. So what changes is that height of the energy landscape. In some of these variants, a structure may be in a bottom, in a well. In another variant, it may be in a peak, and it's still there. You can still use it. The only thing that changes is the energy or the height. So we collected those structures, we stripped away all the side chains, we stripped them to the alpha carbons, a lot of approximations, and then we did the basest, simplest thing that we could do. PCA, principal component analysis, not isomap, because I can't use isomap in order to explore a space, just PCA, principal components. And brought it down to 10 dimensions, was pretty okay, did a lot of analysis in our PLIS paper. The reviewers made us go through a lot of questions, which was good for us. So what I'm showing at the top are basically a schematic of these different MPCs, principal components. The blue dots are the known experimental structures of different forms of HRAS that have been projected down. And then we put together a search engine or an optimization engine that at every moment would basically select a parent and modify it according to the principal components. PCA is linear, but that means it's also highly interpretable. So you can combine the principal components, weight them differently. This gives you the stochasticity, so you can sample new. These are not structures, these are points in the principal component space. But then we would use force fields, and for folks here, an amber force field, we would resurrect them to atomistic structures. So we would bring those points, make them into full atomistic structures with Cartesian coordinates, optimize them slightly, project them back in the space, and repeat this process. Now, each of these portions, select, vary, improve, select, these are different papers. We put a lot of work on how do we think about selection? What is our variation operator? What is our improvement operator? So I'm not going over all of this in the interest of time, but we were able to reconstruct for the first time the full energy landscape. These are dots, structures that we assembled, color-coded by a particular force field, amber FF14SV, for those that might want to read this, kilocalorie per mole unit. The blue regions are low energy regions, so they are more probable regions. And then as it gets to the reddish, think of these as the peaks. But again, these are only dots. I worked with a statistics professor, Dr. Wanli Shao, and we used a lot of topological data analysis in order to build these smoother looking landscapes. This is for the healthy version of HRAS, and it beautifully shows the two basins. And there are even basins within basins here for the two main structures of HRAS that are the open or the closed state. These are landscapes of the oncogenic variants. Basically, the algorithm ran for each of these separately to reconstruct the unique landscapes. And what we noticed with some degree of sort of more clarity than others is that some oncogenic variants completely removed one of the states. So it made them a mountain. So HRAS couldn't get here in feasible timescales because you have to collect a lot of thermal vibrations in order to go up the hill. It was a rigidification of the protein in the always active state, which made sense at a high level because you think, okay, if it's always active, then it's telling tumor grow, grow, grow. For other disorders, we found very different mechanism. Basically, the landscape degenerated, everything went. So it was a protein that could access many different structures. And if you say, okay, what does it mean? I don't know what it means. I'm just telling you what I see. And then maybe experimental colleagues, we have an experimental colleague, Carla Matos, that were studying these systems. Okay. Very quickly now, we were able to actually connect with some experimental measurements because we'll take these landscapes and convert them into vectors or features and then compare, do very simple inference models to compare with experimental measurements. Okay, but this did give us some foundational research as well. We were able to unify robotics-inspired optimization with evolutionary algorithms that none of you care about, so I'm not going to talk about that. We were able to bring some distributed learning ideas because a lot of what we were doing was we were what we call structurizing the search space. So what that means is basically imposing a tree or imposing a graph, right? Putting some structure in a state space. So we used some similar ideas in machine learning in order to distribute learning. So we would have nodes of different learners. It could be like base here, SVM here. How do you organize them? Nearest neighbors, they could exchange training data. We had to think about difficult instances, moving between neighbors. It was a nice result. Nobody remembers PSBML because XGBoost just came out a few months after. And we stood very much well with XGBoost, both in terms of performance and error. But yeah, nobody cares about PSBL anymore. It was all about XGBoost. But I wanted to highlight it as an example of sort of incidental finding, if you will, right? We developed ideas for a particular problem, then we realized, hold on, the same inspiration, we can use it to improve or, to advance foundational machine learning research. Okay, back to generative. When you have lots of data, I have there a little girl making a wish, which is how I feel most days. A lot of the algorithms that we were doing were great, but we had to deeply care about these systems. I developed so much knowledge about HRAS, you would not believe it. You can wake me up at 3 a.m. and ask me about something on HRAS and I'll know it because I read tons of papers. I had to understand a lot. But it doesn't scale, right? All these algorithms for every system, you have to care deeply, you have to understand so that you can say something meaningful. And biologists appreciate this a lot because you say something meaningful and specific, but you don't say something general, right? You don't have really a general methodology. So the question that I started posing to my students is, how can we utilize a lot of data that we have out there, let's say in the protein data bank, to sample the structure space of any molecule? Any molecule, like I don't care if it's HRAS, any molecule. So deep generative learning came easy, or generative AI before it was called that way. But any question that I pose is in its essence a lie. Because any question that I pose has gone through 20 layers of approximation. So the real question that I want for any physicist in the room, given a sequence of amino acids, predict the Boltzmann distribution over tertiary structures, something super hard. All you have to know is super hard. We're not there yet. So I'm like, okay. My students told me, I can never get a dissertation on that. Okay, let's go down. Given a sequence, predict functionally relevant structures. We don't even know what functionally relevant is. How do I capture that? I can cook something up, but I don't really know. Okay, so go down. Given a sequence, can you predict reasonable protein-like looking structures? Okay, we're getting there. But can we make it even easier? Forget about a specific amino acid sequence. Just predict protein-like structures for a sequence-agnostic chain. Meaning, I know these are amino acids, but they don't have their unique identity yet. So that's where we are right now at the moment, brick by brick. So I'm going to skip this. We started just trying to beat a system known as Rosetta. You don't need to know it, we beat it. That's what told me, okay, I guess we can continue in this direction. I'm just going to show you different architectures for those of you that might be curious. We started very simple. A lot of inspiration was from vision. So we looked at three-dimensional structures collected from a database. Think of it as your data set, your repository. We converted them into, they look like images. These are contact maps. So basically we were going back to say which amino acids are close to which amino acids, right? So we're losing the coordinate detail. Once they're images, then we can play and we can see what does your favorite gun do, right? What does your favorite convolutional plus star do, okay? And they all do fairly well. Okay, some better than others. We've learned a lot of lessons. We've learned, for instance, that if you want to generate protein structures for sequences of different length, spatial pyramidal pooling helps. Again, this was an idea in a little noticed vision paper, but we noticed because we were struggling with how to generate different size protein molecules. But the system that we really liked before language models became a thing was graph VAE. I'm still selling on graph VAE, folks. I love these systems. So we would take the molecules and we would convert them into graphs. It's very easy to do that, whether you have a protein molecule or a small molecule, because you have atoms. Atoms become your nodes, and the bonds between them become your edges. So you get your graph. And it has more information than an image, or even more information than a contact map. You can also encode the types of atoms that you have there, the types of edges. For people that know about graphs, you can accommodate a lot of types of features. And then we used VAs, graph VAs. They were very well received. I think first publication was 2017. We played with a lot of variation. Why we really like them is because they tell us something more. Through the latent factors, they tell us what it is we are capturing in these structures. Okay, so we can isolate, we can say only change the first latent factor, keep everything fixed, and tell me what kind of structures do you generate? Because we know, for instance, what we should capture. If these latent factors make sense, there should be something. Maybe the first latent factor is capturing formation of local patterns, the local secondary structure elements. This is knowledge that we have accumulated over decades about protein structures, right? So we want to know, are these systems capturing something meaningful that we know is important when tertiary structures get put together? So this is what this picture shows when we vary specific latent factors, and we found, oh, this one is really first forming the small portions. These other ones are bringing the portions together, but it's not 100% clear, right? Some seem to have overlap, seem to be doing similar things. So we would go and we would peer over, let me see the contact maps that are generated when I freeze all the other latent factors, and I only isolate this one, so I can look deeply and try to come up with an interpretation, right? We wanted interpretive frameworks. The good news is that they gave us the most reasonable looking structures, really protein-like looking structures, and the latent factors were interpretable. So sure, we got a few papers out of it, but most importantly, we realized we can continue in this direction. We can build brick by brick and hopefully go up to that really, the real question that we care about. We look deeply into the quality of the data because we are handed data. This is data by biologists. We have no control over these data. We have structures with missing portions. We have structures with different resolutions. So we look deeply into how data size and quality matter. And this was a very deep, I call it a statistical study that told us these are the parameters that you want if you really want to guarantee quality of data. And it was exciting. The people that was most excited about this were biologists of all, I would say, because we really looked at, the student got sort of a cover in a journal, and they even tweeted. She was happy, it helped her to get a good job. Anyway, we're not there yet at the top, but something else we're doing is we're taking these ideas for hopefully drug discovery, but I always tell folks, if I ever say I'm doing drug discovery, tell me, Amarda, you're losing it. Because we don't do drug discovery. Drug discovery is a $10 billion process, 10-year process with 99% failures. What we're doing is we're playing. That's what we're doing, we're playing. So that's what I tell my students. Do not hyperventilate. All you're doing is you're playing. You're generating small molecules, okay? So how are we generating small molecules? Okay, here's what folks are doing. That's what generative learning is good for. Input, lots of data sets of discarded small molecules that pharma doesn't care about. They're all out there. We have collected them. Can you generate hopefully new novel molecules that maybe, big maybe, can be good leads drug candidates? Okay, the very first question is, can you generate valid things that look like smaller molecules, that don't violate foundational chemistry, right? And that hopefully are novel, because if you are reproducing the training data set, you just wasted all my time, okay? There's lots of work in this space. The work we have done has been, again, we love graph VAs, but something different we're doing from the others is we're doing property control. So the same way that in vision, you can say, well, I want a woman, but I want her with blue eyes, red hair, and a mustache, and glass, you can do whatever, right? That inspiration we got, we said, we don't care about deep fakes, but we care about fake new molecules. And maybe some of those ideas, if we port them over, we can generate molecules with specific properties. It has to have this molecular weight. It has to like water this much. It has to like fats this much. It has to be synthetically accessible, meaning they can actually build it in the lab. It has to have a potential for being a drug. So I'm going to skip over this, just tell you that those latent factors were very important, because they showed that they were correlating not just with molecular geometry, but also with molecular properties. Some of them were capturing molecular properties. So we realized if we control the latent factors, we can additionally control for properties. And we have come up with different sort of architectures. I know this is a little bit hard to parse. The idea is that we are in the latent space, we are reserving a portion, this W here, to capture the properties, okay? And so during the training process, we think, do we do that explicitly? Do they do that implicitly? This is all those dashed lines, those dashed arrows, different models over the others. And we have pretty good, I would say, control, but not quite monotonic control. Because it's hard, when you make changes to graphs, they are discrete. But the changes in properties and monotonicity, you want continuity. So that is the relationship that we're trying to still parse, and that is a growing area of research. Okay, so I'm going to skip over this. Final word. We want to go back to discriminative, but the question is, how do you incorporate lots of data in a discriminative setting? And I was telling our faculty today that we were doing language modeling research, but I was calling it sequential models, right? Like we started with RNNs and things like that. What we want to do in my lab now, we're studied, we want to leverage knowledge. I don't want to go away from the domain insight, understanding of a problem. Well, partially because I can't build more than 1.4 billion. That's how big my student was able to go last week, 1.4 billion model. And beyond that, he said, nah, it's going to be months and I'm not going to be able to catch any deadline. So how do you catch up maybe to the transformer, potentially with alternative architectures and potentially by leveraging additional knowledge? So you have seen the size explode. Not a big believer. I don't think they're good for the planet, energy consumption, just read stuff about data science. I'm very biased about this, I understand. But I think we need lighter models. We can get to lighter models, we can situate them. They don't have to be foundation models. I'm okay with leaving that to Google and OpenAI, but models that work, that help us to advance particular scientific problems, we can situate them in specific knowledge and structure them so that we can get to even not just catching, but beyond the transformer architecture. So foundation models, they actually are not doing very well. I'm going to show you. This is a very classic problem in molecular biology, remote homology. Basically, it says two proteins, they look very similar, their sequences are very different. If I give you those two different sequences, you won't be able to tell me that the structure is the same. Okay, so here we have evaluated different ESM1, ESM2, we limited at 650 million, ProTrans5 is 11 billion parameter, but we made the game difficult. Because what they publish in papers, they publish stuff here. In their data, they allow proteins with a high sequence identity. They're not the real challenging problems. So we brought them down to 20% sequence identity. We call this the twilight zone. This is the real difficulty for remote homology. And they're all pretty, like you can't tell that, oh, this is ESM and oh, that is ProTrans. So we haven't solved remote homology for all the work on going big. Variant prediction, so telling me when there are mutations, is this pathogenic, is this a rare mutation or not? Again, we haven't solved it. This is a little bit hard to decode, but the point I want to make is that they are no better than classifiers, sweet, good old classifiers or conservation-based methods. We can really not tell that they are better. There's a lot of overlap in their performance. So again, it's like my grandma used to do oop-de-doo. Like, what did you do with the 650 million? I don't know. For variant prediction and remote homology, not much. So what we're trying to do is putting back more data. So I don't need 650 million, maybe a few million. I can integrate in my sequence structured data by focusing that attention mechanism with those contexts and do as well as or better. I can put in functional information like Go ontology. Again, I can put different biological knowledge. I can leverage cross-attention to bring back physics into the problem. So I can predict, for instance, transcription factor binding sites and other very challenging problems. So the point of this is we're doing a lot of work in trying to bring other sources of data or sources of knowledge because we cannot afford large models, and plus it's not obvious that really progress can be made by scale alone. We just organized a workshop, the AAAI, because I am trying to push large language models, or largish as I call them. Largish is like a few billion parameters for us, okay? But there's a lot of work in the lab on bringing ideas from those energy landscapes into how to actually do optimization for deep learning, because you end up with one model, but is that the model that generalizes better? I don't know. So maybe bring back some dynamics into the kitchen. And then I have a student that is trying to investigate, it's going back to recurrent neural networks or a form of them, state space models. Birdie calls it, he loves birds. And he wants to figure out whether they're better than transformers. And this picture here is supposed to show that indeed Birdie and variants of Birdie can catch up to the attention on many natural language understanding tasks, so beyond biology. So the point of this is it's important not to follow the crowd. Okay, try to do something different. These are all the people that I'm highlighting. There are many more I couldn't fit in this slide. And I believe I am done.